Hello everyone. So this semester in Genetics Lab 312, we investigated the fitness of budding yeast in varying salt types and concentrations while looking at two strains of yeast. These two strains were the domesticated BY4743 and the wild VS189A. These photos are of our second spot plate experiment 1 week after plating. The details are contained in the legend below. To quickly summarize, the W indicates a row of wild yeast and the D indicates a row of domesticated. As you can see, one of the most interesting results occurs in panel C, which is a YPD plate with the addition of 0.8 molar sodium chloride. The wild strain has been wiped out while the domesticated strain has survived. This indicates that the domesticated yeast have had a more successful osmotic response in the presence of sodium chloride. We wanted to find the genetic difference or differences between the two strains that could potentially explain the disparity in osmotic response. One logical place to start is the HOG1 osmotic stress pathway, and then look for genetic differences connected to this pathway. So what is the HOG1 pathway? According to Stato and Posas, when yeast experience hyperosmotic stress, they undergo biological changes such as the temporary ceasing of the cell cycle, modifications to normal transcription and translation patterns, and importantly, the synthesis and storage of glycerol, which is an osmolyte. These changes are essential to survival in hyperosmotic conditions and are largely governed by the high osmolarity glycerol pathway, abbreviated as the HOG pathway. That sounds important. So how is the HOG1 pathway activated? The HOG1 response pathway begins with the detection of high osmolarity conditions. Osmosensors are intracellular receptors that detect these changes in the extracellular environment. In the presence of high molarity conditions, the osmosensors recruit a signal protein that will undergo post-translational modification. To skip to the end a little bit, since HOG1 is the last step before cellular response, it is classified as a MAP kinase. PBS2 is the step before HOG1, making it a MAP kinase, or MAP2K. You probably guessed that makes ST11 and SS2/22 a MAP3K. The step before the phosphorylation of the MAP3K kinase can be a MAP4K, or the signal molecule that was originally recruited by the receptor. This process is referred to as a MAP kinase cascade. A MAP kinase response of signal amplification results in the activation of the HOG1 kinase that goes on to induce cellular modifications. As you can see, the activation or phosphorylation of HOG1 leads to several intracellular changes, such as modifications in transcription. HOG1 acts as a transcription factor, which leads to numerous protein-based changes that facilitate the yeast response to osmotic stress, such as the production of glycerol and the halt of the cellular cycle while adaptations occur. So in summary, the HOG1 pathway leads to cellular changes that help the yeast survive in conditions of osmotic stress. And there are the references.